Chapter 7, Patterns of Inheritance. Have you ever wondered why you look like your mom but not your dad? Have you ever wondered why when your puppy has little litters or your kitties, that they look like one parent but not the other? Or maybe they have traits in common with both? That is what we'll be talking about in this chapter. Sometimes, if a tall man marries a short woman, you get all tall boys. But sometimes you get a mixture of them. Why is it sometimes but not others? That is the subject of this chapter. Genetic trait is a characteristic that you can inherit. There are physical traits, there are biochemical traits, and there are complex traits that are behavioral, like a dog's shyness. The gene is the basic unit of information affecting a genetic trait. Now, one gene is one polypeptide, one gene is one protein, but it's also one trait many times, but not in all cases. A chromosome contains many genes, and that gene is the code for a single trait. Genotype is the genetic makeup, phenotype is the physical. Here's how you can remember that. P in phenotype for physical appearance, what you look like, hair color, eye color, personality, maybe even your biochemistry. Are you lactose intolerant? That's an enzyme phenotype. Genotype is your genes or your letters if we're talking about alleles. You'll see what I mean by that in a minute. One is dominant to the other. That means the dominant gene or the dominant allele masks the expression of the recessive allele. Now, what do we mean by that? What we mean by that is that every gene has a letter that goes with it. You have uppercase letters and lowercase letters. The letter comes from the first letter of the dominant. That is usually how that works. The first letter of the dominant trait is where the letter comes from. So, for example, if black fur allele, which is that version of the gene, is dominant, you use a capital B for that black fur. So that means the brown fur would be a lowercase b. That's not a good example because they're both beginning with B. For example, let's do eye color. Brown eyes dominant to green. The letter you would use would be the B, not the G. Homozygous means you have the same, either both big B's or both little b's, and heterozygous means you have one of each. Mendel is the father of genetics. He invented the field that we know as genetics. How did he do that? He worked with pea plants and he crossed tall plants with short plants and got tall plants, and yellow flowers with white flowers and got yellow flowers. And he came up with these ideas of dominant and recessive. Those are called genetics crosses. When you cross a tall plant with a short plant and get all tall plants, that is the P generation generating the F1 generation. When you self-cross, which means you take those plants and you cross them to themselves, sort of like inbreeding, but plants do it all the time, that is the F2 generation, and that's really where it gets interesting. The F2 generation is where it gets very interesting. To figure out the F2 generation, you have to draw a Punnett square. On the left, you put the eggs, and the top, you put the sperms, and then you bring the letters down, and you find out that you get big P, little p twice, and then in this little box down here, you get little p, and then you can do the math. 75% or three quarters of them are purple, which is the dominant, and only one quarter or three in the case of a 3-to-1 ratio is the recessive. That is a classic monohybrid cross. You can determine it with a Punnett square. This demonstrates two laws of Mendel. The law of segregation, when two alleles of a gene are separated. Now, you know when that happens because you've drawn meiosis and you've drawn mitosis. When you separate your chromosomes in anaphase 1, that is the law of segregation. Probability, you will be answering questions in terms of probability. What's the probability that if a tall and a short mate, that their offspring will be short? In Mendelian genetics, it's one of those boxes out of four. So it's 25% or one quarter of a chance. For example, in a genetic cross between two heterozygote black-coated dogs, the probability of getting a puppy is three quarters or 7.75. The outcomes are never certain unless true breeding individuals are crossed. That means dominant or recessive with the same exact letters. Another law that Mendel discovered is the law of independent assortment. Now, again, he didn't have the advantage of knowing what you know, and that is about how mitosis works or meiosis. He didn't know that because the microscope and chromosomes had not yet been discovered. So he was doing it all with math. But you know that independent assortment means when you draw your mitosis and your meiosis drawings, there's different ways to line up at the metaphase plate. You can be red, 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 blue, 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 or it can alternate. And those different ways of lining up allowed there to be independent assortment of those genes and of those chromosomes. Here's a dihybrid cross, very complicated. It involves two traits. That's why you have four letters, two R's and two Y's, crossing with two more R's and two Y's. Dominant here, recessive here. And what do you end up getting? You end up getting all dominant looking. They look like the dominant parent. When you cross them to themselves in the F2, you get this huge Punnett square, which breaks down into a 9 to 3 to 3 to 1 ratio. Nine are double dominant, which looks like this parent. One of them out of 16 is double recessive, that looks like this parent. And then the threes are alternating combinations of traits that look like neither parent. That is a dihybrid cross. It involves two traits in which you are a hybrid at both trait. Mendelian traits are controlled by a single gene and unaffected by environmental conditions. Mendel basically worked with 7 traits that all behave this way. He either got lucky or he threw out the data that didn't behave this way because it turns out genetics is way more complicated than those 7 traits. You know that if a tall and a short marry, they have middle-sized kids and everything in between. The chromosome theory of inheritance basically says what you already know, and that is that genes are located on chromosomes. Chromosomes come in pairs, one from your mother and one from your father, and they separate during meiosis and then recombine during fertilization. That is called the chromosome theory of inheritance. Incomplete dominance now is a different way of expressing dominance. When you have true dominance, Mendelian dominance, when one allele is dominant to the other, that is called complete dominance. Not every genetic system behaves that way. For example, if you take a red flower and a white flower in snapdragons and you cross them, you don't get red, you get pink. All right, so that's called incomplete dominance. A heterozygote displays an intermediate phenotype. Look at these two dogs, one extreme, the other, and then you get something in between. Codominance is different as well. That's a third system, and in codominance, the effects of two alleles are equally visible in the phenotype. In other words, they're polka dotted. So instead of getting a red or a white or a pink, you get both. A good example of that is blood type. Blood type A, blood type B. When you put them together in terms of an AB genetic individual, you get both. They're polka dotted. They're both A and B, and the blood type is said to be AB. Complex traits, a genetic trait whose pattern of inheritance cannot be predicted by Mendel's laws of inheritance. They don't fit the single straightforward, single gene, single phenotype already discussed so far. Pleiotropy is an example of a complex trait. It's when a single gene influences many traits, such as albinism. You can be albino at many phenotypic areas, your hair, your eyes, your skin, and yet it's a single gene. Polygenic traits is when a gene is covered by many genes cover one trait. Most of the famous examples that we like to talk about, eye color, hair color, are polygenic traits. A gene can have more than 2 alleles. This is called multiple alleles. An example is the blood system. I know what you're thinking. You're saying, wait a minute, I thought you said that was codominance. The relationship between A and B is codominant, but when you have an A allele, a B allele, and an O allele, that is an example of multiple alleles. And they have to understand how each of them interacts together. In the case of the ABO blood type system, A and B are codominant, but each are dominant to O. So an AO individual is blood type A. A B and an O in an individual is blood type B. The only way to get blood type O is to be double O, OO. Then you would be blood type O. Epistasis is yet another complex system. It's called non-Mendelian and it's complex. It means that two genes are inherited independently, but the alleles of one gene affect the expression of the other. For example, there's two genes, a B and an E. The only way the puppy can be black is if it has a dominant B and a dominant E. If it does not have that, it will either be brown or yellow. That is epistasis. Another way to tell epistasis is if there's a 9:7 phenotypic ratio. If you did a cross, instead of getting the 3:1, you would get a 9:7. That's a key giveaway that it's epistasis. But making traits even more complicated is the environment. Sometimes you'll have a gene, but it will be not shown because the temperature is too hot or too cold. So there's environmental factors that play a role. You could also have CO2 levels in the amount of sunlight. Those are all examples. Siamese cat coat color is an example. The temperature depends and creates the pigment that gets expressed. Most chronic diseases are complex traits. It's not as simple. Mendel either cheated or he got lucky to get those seven traits that were just tall and just short. You know, in real life, genetics is very complicated. A disease can be genetic, but it can also be influenced by other genes and other environmental factors. And maybe the gene mutates throughout your life or the gene turns off. That might make it complicated as well. Common diseases in industrial countries caused by multiple genes interact in complex ways. For example, heart disease, cancer, and stroke. Very complicated, not simply a matter of you have the gene for heart attack. It's much more complicated than that. A major goal of modern genetics is to identify genes that are contributing to these diseases. Someday we will know what gene causes every trait, and we will be able to do gene therapy, gene surgery, and fix the gene in each person. That still remains the holy grail of medical research. Here's something that's really fascinating. Sometimes a complex organism, like a human being, that is high intelligence, complex behavior, you would think that they have the most number of genes. That's not true. It turns out lungfish has way more genetic material and way more genes than we do. It's actually shocking. Plants are like that too. They have many more genes than we do. It's very fascinating. And that is chapter 7, Patterns of Inheritance.